really thank you all for being here for our Botanical Safety Annual Meeting. I'm Connie Mitchell. I'm a Senior Scientific Program Manager at the Health and Environmental Sciences Institute, or HESI Global. Myself and Julie Shikva, who's on camera still, manage the Botanical Safety Consortium. But really, it's on behalf of this large expert group that you're going to hear from many of our panelists that really help lead the different teams. So we're really excited to present a lot of different information to you today. So here's an overview of our agenda. I'm going to go through the background information and some high-level updates across the teams or things we won't get to in the individual working group presentations. But then we're joined by a really nice panelist of speakers that are active on the working group and lead them, that are going to say what the different teams have done over the last couple years, and then really emphasizing what's been done in the last year to move forward. So I think it's going to be a really exciting day of talks showing different assays and the botanicals we performed in them. Oh, sorry, one last thing. And then we will wrap up. Julia will help do some wrap up questions, polls, get audience engagement from you all. And then we'll have time for questions if there are any that cut across. Okay, so I'm going to start with some intro. Folks have seen some of these slides before if you've come to our other meetings, but I think it's really important to get us all on the same page. So this is really a toxicology project, but it's about botanicals. So what is different about botanicals compared to single chemicals? So plants are chemical factories. There's lots of different plants. Really, our definition of botanicals is a little bit broader, even from, including things like fungi and other things that produce products. But really, we're looking at quite a few plant species that have been used for medicinal use. We know that their phytochemicals can exhibit a broad range of bioactivities, and that humans have been utilizing botanicals and plants for a long time for medicine, for pesticides, for cosmetics, consumer products, poisons, lots of things. And it's really a spectrum of going from, the single chemicals within the botanical versus using the botanical itself. Oops, sorry. And I really like this slide. This came out of Kristen Ryan's work at NIEHS, but I think it's a really nice illustration of why botanicals are complex. Unlike a single chemical that you have a lot of confidence in what you're testing, there's a lot of steps along the part of like going from the plant itself to going to the human exposure that can change the composition of a plant. So, if you're growing a plant, it really depends where you grow it. That might affect the chemicals there. If you grow something in India versus Sri Lanka, what part of the plant? So is it the root? Is it the whole plant? Is it the leaf? Was there something different that year in the climate? When it was harvested, was there something else in there? Were there other plants? Was there soil? What about when you get to the processing phase? We talked a lot about in the consortium about what type of solvent we want to use to extract it. That'll really change the chemical composition, what chemicals you're pulling out, and the kind of the manufacturing process. Is it the same process every time, or is it slightly different? What about other, things that are potentially there that aren't on purpose, adulteration or contamination issues? What about storage? Is it stored for a long time and maybe in light and things start to degrade? Is it stored frozen? So that'll really change the chemical composition. When you get to the product level, a lot of these are combined with other botanicals. Sometimes they're in pill form, so the excipient will change how you're exposed to it. Again, other things that might happen at that phase, such as contamination or adulteration, will change it. And then the human exposure, and this is similar to, for single chemicals, but are people taking the right doses? Are they taking a lot? Are they taking it once, 1 week for a cold? Are they taking it every day for their well-being? Do they have some kind of genetic disposition? So there's a lot of things that change how humans are exposed to botanicals. And dietary supplement use, we're going to talk about a couple different classes. This really started as a dietary supplement project. So that's something, of course, we want to look at and how the use of that has gone up over time. So looking the last 10 years, we see, almost a doubling of sales. If you notice between 2020 and 2021, it's a really big increase. There's a little bit of leveling off, but still there's this is a big market. People are taking these products. There's really this desire for natural for consumers. So there's definitely the exposure. And people use it in kind of for wellness, but people also use it, of course, for herbal drugs. So there's, reports in sub-Saharan African, but it's not specific just to, Africa or developing countries. In Germany, people use our herbal medicines. People use this all around the world as really a desire for natural, for things that they perceive as safe. Same there in Peru. And then, you're going to hear today about our dermal working group. So people are also using this in cosmetics. That's been happening for thousands of years, but again, there's this kind of renewed interest on using naturals in cosmetics. And so while it's uncommon, there is instances of toxicity, and we want to make sure we're protecting our consumers. And so, one of the most famous example that we talk about, and we'll talk about ephedra today, is the ephedra alkaloids. People were taking ephedra and hydroxycut for weight loss, and that really, led in some cases to death and some issues. There's lots of people in the consortium that has worked on this. Sometimes it was more with combination with caffeine, but still, these are banned in the US because they present an unreasonable risk, and that's almost 20 years old now. So you still see products like hydroxycut, but now you see ephedra free. There's also a lot of issues with maybe swapping what the botanical is. So there's some issues of people taking a root from Mexico that they thought that's what it was, but it turns out it was yellow oleander, which is a known poison. So there was some issues pretty recently of people taking these supplements, and really it was a case of either intentional or unintentional swapping of botanicals of the plant itself. And so this again led to really adverse effects, I believe in some cases death. And even things that we know really well, like green tea, if I like to drink a couple cups of that a day, but if you really concentrate it, there's issues that might happen. There's a report led by USP, and then there's also this EFSA safety assessment that if you have this really concentrated EGCG, there could be a warning for specific, people with either susceptible conditions or if you haven't had food, but there could be potentially liver toxicity again in this really concentrated formulation. So we have these different examples. We'll show some other examples today of ones we selected to test in our assay. But again, this isn't, just a totally theoretical thing. We're seeing adverse effects. So we really have a duty to the public to make sure these products are safe. And so that led to the formation of the Botanical Safety Consortium. You'll see some of the folks today will be speaking, but came up with this paper a few years ago in 2019 about forming this really multi-sector, people from industry, from government, from academia, coming together to modernize how we look at botanicals. And after that, a few years later, or that same year, excuse me, a few months later, there was a statement from FDA Commissioner Scott Gottlieb on the agency's new effort to strengthen the regulation of dietary supplements by modernizing and reforming FDA's oversight. In that same announcement, he announced the Botanical Safety Consortium as a public-private partnership that will gather leading scientific minds from industry, academia, and government to promote scientific advances in evaluating in evaluating the safety of botanical ingredients in dietary supplements. And importantly, mixtures is there. So that's really what we're focused on. You might hear some work today about constituent like quantification or in silico work, but all our work in the assays is going to be on the mixture itself. Later in 2019, we officially, my organization, the Health and Environmental Sciences Institute, was named as the convener. This was in a memorandum of understanding with NIEHS, part of NIH, and also the FDA. And so this was a couple years ago, and we're pleased to announce that as of, I believe, August, our memorandum of understanding has been re-signed with these two agencies, even with everything this year going on. And this extends for another 5 years. So we have this memorandum of understanding for at least 5 more years. And so what are we doing in the Botanical Safety Consortium? What we're trying to do is evaluate the suitability of toxicity assays for botanicals as complex mixtures. So what methods that already exist, which new approach methodologies can we use for botanicals as complex mixtures? Are they ready to go as is, or do we have to adapt them for these complex mixtures? So that's what you're really going to see in the different working groups today, presentations of the work that was done there. And what we're trying to figure out is, are they okay for botanicals as complex mixtures? Are there special considerations? Are they picking up the positives and negatives we think they are? So you'll really see a lot of that today. So I think a lot of folks know that funding this year has been a bit of a bumpy road. Prior to this year, the FDA and NIEHS provided about 60% of the budget for the consortium, and the rest matched for industry. I will say we really thank our industry supporters. So a lot of the companies, and I'll show them in a moment, involved pay a rate to be a part of the consortium. And this has really been what allowed us to keep going when we lost our federal funding unexpectedly this year. And additionally, we really went back to our members and explained this kind of gap in funding. And four companies came back and were able to give us additional funding in addition to what they already give. So this was really a stopgap that allowed us to keep going. And then I'm really pleased to announce that as of October, the federal funding from NIEHS and FDA has been restored. So we're really back in good shape, but it was essential for the companies that helped us kind of get through that rocky time where we lost our funding for a few months. So I just really want to thank you all. And you also see as we present a lot of the research today that yes, there's actual dollars involved, but a lot of things that done for the consortium are in kind, both in expertise and people volunteering their time, but also a lot of the experimental work has been done in kind or at cost. So I just really want to thank you all helping us through this year. I think we're back in a really good shape and position now. So with that, I want to thank our members. So we have really good participation across sectors. This is really essential for all HESI projects that we have people involved from government, from academia, and I'll show in the next slide from the private sector. So we have different regulatory agencies involved, mostly in North America and a little bit in Europe. You know, this is something we'll call out to all of you today. If you don't see your name on here and would like to be, it's a standing action to reach out to me and Julie and see how we can get you involved. We have lots of different academics involved in research institutes. And this is where a lot of our research has taken place with some of these cutting edge assays that people have in their own labs. And then we have quite a few industry members. It's a really nice mix of different kind of assay labs or chemical analysis labs, some of the bigger companies that work in this space in the dietary supplement or natural product space, some of the other trade associations that really have a lot of members that maybe are smaller and are here. So we have a really nice mix of industry members involved in the consortium. And then for other scientific organizations, we have some consulting groups, we have some other kind of scientific groups that really participate and have helped us. So this is, you know, I'm presenting today. Me and Julie get to present a lot on behalf of the Botanical Safety Consortium, but it's really because of the members we have that we're able to do this. So just thank you to all of you. And so I really like this slide. Michelle Embry created it, but I think it really nicely distills just the amount of work we've done in the last couple years. So you know, we'll go into our sixth year in 2026. So we've been active for a couple years. I think we're really at a point where we're starting to show, you know, a lot of manuscripts, a lot of data. And so it's a really exciting time that took quite a lot of careful planning to do well. Really, the driving force is our technical working group. So we have a lot of different members across the different teams that I'll show in a minute. And with that, we also have folks that are maybe ad hoc or stakeholder council members, and we go to them when we have specific questions or they get occasional updates, but they really are still essential members of the consortium. We have about 50 different organizations, those the ones I just listed. What you'll hear today is potentially different numbers of botanicals that we've tested. We started with 13, and these, you know, we'll go into why these were chosen and what they were. But then we decided we needed some specific for other endpoints as those working groups were added. So we added 3 for the neurotoxin cardiotox. And then what you'll see later today is the 9 we added for dermal, and those are newer. And we're looking at different new approach methods. And thanks, Julie. Julie's going to put some links in the chat too. So I encourage folks to look at that. So we have, I'm going to say about 18 new approach methods we're looking at. Why it's plus and not an exact number. There's some, you know, in silico tools we're looking at, again, on the constituent level. There's some that are multiplexed together, but these are really the different like endpoints and assays we're looking at. And we've really received this nice mix of federal funds and private sector funds, But I really emphasize the amount of work we're presenting isn't just based on those two amounts. It's really based on, again, a lot of contributions from folks on the team. So we'll highlight that as we go. We have 8 different groups that meet monthly. I'll show that on the next slide. And then we have quite a few publications. This has really been a banner year, so it's exciting to show that. I'm going to show a few, but most of those will be discussed in the working groups. We have a couple under review, and we have numerous in preparation. So with that, these are our different working groups. We have them divided into kind of different sections, but there's of course overlap and interplay between them. So we have our different toxicity endpoints. You're going to hear from all of them today in presentations on their teams. We also have some ones that are more focused on exposure or cross-cutting issues. So we have ADME. This team is really looking at different tools right now, mostly in silico, but how that information could support the interpretation and evaluation of some of the in vitro results. We look at botanical drug interactions. This is mostly housed in our hepatotox team, but this is really an important consideration. We have a chemical analysis team. I'm going to show a few slides on that today since we don't have a full presentation from them. Data analysis, who have been really helpful in both planning and interpreting the studies. And then pharmacognosy, who have been really essential for helping us, pick and source botanicals, helping us with analytical methods, and really an understanding of how these botanicals are taken traditionally, modernly, etc. So we have a lot of really active groups or themes in the consortium. And this is our overall strategy of how we picked our endpoints, how we picked our botanicals and assays, and where we're trying to go. So what all the different toxicity groups did, like hepatotoxicity, is they went through the literature to look for data-rich botanicals that have a lot of information, which, compared to like pharmaceuticals or ag chem, is a lot harder in different formulations and all the things I already emphasized. But to pick ones that we're fairly confident on can lead to, say, hepatotoxicity or do not. So it's important to have some negatives too. And they selected their list. We combined this list across groups. We really tried to pick ones that maybe hit multiple endpoints if possible. We then worked with different labs on chemical analysis evaluation. I'll talk a little bit about that on future slides. In parallel, what the groups did too is identify assays and approaches that are used for single chemicals that we want to apply for these botanicals as complex mixtures. For some teams, it was, I'll say, somewhat easier, like for genotox and for dermal, since they have a lot that have OECD validations. For other teams, we were really trying to select, what do the folks on the team have in their expertise, what can be done, and what's accessible to many groups. And with that, we planned the studies and we ran the botanicals through the different assays. You're going to hear a lot about that today. We're really in kind of these two boxes. So we evaluated most of the botanicals. We're really working on analysis and understanding our results. And where we'd like to get to in the end is have a toolkit of assays that others can use. This might be, maybe we eventually get to the point where this is a step how-to framework, but right now this is really more, what tools are out there, what work well for botanicals, or what might need to be adapted. So this just gives a snapshot of the initial 16 botanicals. So these, lots of different numbers floating around, but basically the ones we picked initially, then the 3 added for cardiotox and neurotoxicity. And how we selected these was based on toxicity information. And so this is really looking in the literature to see, what human data existed. And this could either be, a clinical trial or in a lot of cases, adverse event reporting. This could be in vivo evidence. So there were a lot of nice studies from the National Toxicology Program we were able to utilize in other peer-reviewed literature, and then in vitro evidence. And in most cases, it was a combination of these. It was also nice if we could know what the, bioactive or toxic constituents were. One thing is, are we able to source them? So some were suggested, but we really didn't have a good source on. And do we have a way to quantify the key constituents? So these are the different ones we selected. We'll hear about them from the different teams as case studies and why. And maybe just highlight, one or two. So for example, Aristolochia is one that I don't really think people take as a dietary supplement. I hope not. It has aristolochic acid, and it's a really nice example for gene tox because of those aristolochic acids. Another example could be Asian ginseng. This one was selected as thought to be a pan-negative across groups. So again, this is one that it's really important that we have some negatives to kind of look at our responses and see how the assays are performing. So this is, again, a big table, lots of information, but gives you just a sense of the magnitude of the different assays the groups are looking at. Some of these are done separately, some of these could be complex together, and they're just in different statuses. So a lot of them we're still looking at the data in the different group. For some of them, there were maybe, methods or assays, excuse me, like in the guideline that tell you how to analyze the data. So it was a little bit easier. For others, we're still digging into the data, and there's a few, that we would like to do in the future that have risen on our list since the last few years when we initiated a lot of this work. So you're going to hear about some of these assays today in the different presentations. Not all of them, though, just for the sake of time and not every botanical, but really to give you a flavor of what's coming. And then also to set up, a lot of the papers within the different groups that we will be really trying to work in the next year or two to get this data publicly available. So what are we going to do with the data? So when we get the data within the assay itself, we want to understand, was it valid? Were the controls okay? What were the differences compared to controls? And then we really want to benchmark to the literature calls we made. So if we say, again, aristolochic acid and aristolochia is positive for gene tox, did we get that in the assays? Can we benchmark? We're focused on screening tools here, so that's another thing throughout. There's probably many, many steps that would need to be done to get these tools ready from going from the screening level to a risk assessment level. So really here, can we almost do like a hazard screen? We also want to make sense of the results. So we all know that an in vitro positive doesn't necessarily mean it's going to be toxic in a human. There's lots of other things to think about, like concentrations and the ADME properties. Something in vitro, it's a higher level of exposure and it might not be absorbed by a human or might be metabolized. So really trying to keep those thoughts in mind and contextualize them with toxicokinetic information. What we would like to do once we have some confidence on certain assays and can make, recommendations, either can they be used as is, are there things to think about when running the assay for complex mixtures? Others could then use it for their different scientific questions. So maybe this is screening, quote unquote, new or data poor botanicals. Maybe this is taking 10 different of the same botanical, 10 different lots of green tea and comparing them. This would hopefully help us deprioritize testing for botanicals in animals. This is something we're not doing in the consortium, testing on like rodents, traditional toxicity testing, but this does still happen. You see it in the literature. So hopefully these new approach methodologies could help us, better prioritize which ones don't need that testing, or if they're in cases where there really needs to be something, make it much fewer animals. Also, a lot of these assays give mechanistic information, not all of them, some are more functional, but it could help us fill knowledge gaps. So we really hope that once these assays have been evaluated for these different endpoints, we could then turn it over to others. So I have a few updates. I'm looking at time. I won't take too long on this, but we don't have a presentation from our chemical analysis working group this time. Next year we definitely will because they have launched more work on our dermal team. But just want to really highlight how fundamental this team has been. And with the 16 I talked about, what we really wanted to do was make sure we're testing what we think we're testing. And so we did different methods to try and make sure our key constituents that we want are there, that, using things like HBTLC, that it compares to other reference lots and it is what we think it is. And in some cases where we had larger amounts, make sure there's not contaminants. And this identification and quantification really helped us do in silico modeling, both for toxicity and for ADME. So it's been really important. And Julie just put in the chat, she's very on it. Thank you, Julie. This data is public. And so the work we've done for the chemical analysis is on NIEHS's database, chemical effects for biological systems. And it's citable. So it has a DOI, which is really nice and makes it nice for the papers. This first one, sorry, my mouse is on the other screen. It looks at the botanical constituents quantifications as an Excel table, so it makes it really easy. But then there's also information on the individual botanicals themselves. That we also have a paper that Saramia, one of our co-chairs, along with Vinsika, led for this group to really say, how did we do this? How did we get the botanicals? How did we prepare them? And so with a lot of these two, almost all of them for the initial set are 95% ethanol. This work was done at the University of Mississippi, and that really allowed us to then get to the bioassays after we got the information on the chemical analysis. I want to just give a few updates since our last annual meeting. A lot of those talks, what I presented before, we've heard before if you've come every year. One thing last year, we got to be in person at NIEHS in North Carolina about a year ago. So we had about 40 of our working group members and steering team together. It was really cool to see the different presentations on one of the days and then dig into the data on the second day. That really helped us kind of plan what to do with the data sets that you'll hear about this year. We also have launched a special issue for the consortium. So Julie put the link to our website that has all our different papers. So far, they've gone into different journals, which we can continue to do, but we are having a special issue that was really led in conjunction with Igor Koderbash at University of Arkansas Medical Center in Pharmaceutical Biology, where he's the editor-in-chief. And so this is specific for the consortium. It's not just all botanical safety, but it's a really nice way that if the working groups agree, we can put some of our papers there. Again, the working groups can decide where their paper goes if it's better in a specialty journal for that endpoint. But we have 3 papers that were accepted in this last week. They're not online yet. I can't share a DOI or a screenshot of that yet. But some of these I really encourage you to look at for. One is a summary of what we've done the last 5 years at a higher level, so from vision to reality. And it really summarizes, a lot of the decision making, where we are with the different teams, what have been the challenges and successes. So it's a short paper, but really will kind of encapsulate what we'll hear today. But it was from the summit last year, so there'll be more information. We have 2 other papers, one led by Remco Westerlink. It's some of the work he's done in his assay looking at MIEs for the neurotox group. What's exciting about this one, it's both a concentration response, but then in a few cases, he was able to compare to individual constituents. So for example, we tested aconite as a complex mixture, and then he also tested acatinine to try and look at the differences. And then one paper that was accepted, I think 1 or 2 days ago, that I'm really excited about is part of our global outreach. It's a study we funded with investigators at the School of Pharmacy at the University of Malawi, and they did a survey to look at the potential interactions of people taking herbal products and pharmaceutical medicines to see if there's any potential herbal drug interactions. So that paper is also going to be out soon. So I encourage you all to, look at our LinkedIn, our website. There's going to be quite a few new papers soon. I will say too, and you'll hear about this in the team, but every working group has drafted their strategy paper. This is a really nice way that we can lay out what assays we're looking at, and then what botanicals we selected and what's known about that botanical with respect to that endpoint, like cardiotoxicity. So that one was accepted again, maybe a week or 2 ago. It was pretty soon. Happy to get that out for cardiotox. Gene tox as well. I think you'll hear about these in the individual groups. So I'm almost done. Just a note too, we have different papers for the different teams, but we don't have presentations this time. So Vincinca led a paper on ashwagandha, and this is really using a multi-detector approach to look deeper than we did for necessarily all the botanicals, but really to quantify a lot of constituents. I think it was 30 or 40. And so this one we did a deep dive there, and it was really on how you could use this multi-detector to get there. So that's out. I will just say all our papers are open access, so really trying to propagate the info. And then Yiting Liu, who sadly can't be here for the FDA and government shutdown, but she's done a lot of work on the in silico side with ADME to look at how these tools can be used for botanical constituents. So she looked at some methods used for pharmaceuticals, like the extended clearance classification system, and how that might predict how the botanicals move through the body. We did a lot of outreach. I think you're going to mostly hear about this in the different teams, so I won't steal thunder. But just note that we really try and make it a part of our mission to propagate the work we're doing, both at a high level, kind of like I'm doing now, but also the specific research. So last thing I'll say, as you listen to presentations today, I hope it sparks some ideas. Each group is reviewing their data, But I would say we're open to additional analysis. We would, of course, run this to the group and to the steering team. But if there's things you're thinking about or comparisons that we'd like to make with these data sets, I think it's a time to start thinking about, what else could we do with these data sets? Are there other ways to analyze these data? And even once we make the data public, hopefully again in the next year, we're still open to collaborating. I think I'll end there. There's our website. You can get in contact with me, Julie, the general email. Please ask questions today. We'll have time after each one. But I think with that, I would ask if folks have any general BSC questions to put it in the Q&A and I'll take a look. But just so we stick on time, I think it's time to move to Christine. So I'm really excited to introduce Christine Witt. She had been at NIHS working on gene tox for both single chemicals and botanicals, and she is one of our co-chairs for our genotox working group.